Huh.
defect is occurring both during the embryogenesis, so in the womb, and also after the fact. And then I'll just say the secondary way we confirm this is we also gave a tenth of the amount of fructose, so even less amount, directly to those neonates. So during an early day 2 to day 4 post birth,
Yeah.
if you just give them one tenth of the amount of fructose into the stomach, you still see the same exact phenotype giving it to the neonates. So even less fructose, if you're young developing
Wow.
children are doing this, there's still that effect. So those are the sort of experiments, big ones.
Wow. So fructose exposure during pregnancy, in the womb, or afterwards, even if it's quite a small amount, is causing this failure of phagocytes to clear out stuff in the developing brain. So then there's too much stuff in the developing brain.
Absolutely. Yeah.
Wow. What comes next in this story?
Yeah. So, I mean, this is where we were curious if this was the, classic, is it cell intrinsic to the microglia or is it something happening on the outside? And so we, through a series of experiments, we basically made new mice, a whole new mouse strain where we could specifically delete the transporter that I mentioned, GLUT5,
Yep.
from microglia. Actually, it deletes it from all macrophages and only in the neonates, so in the
Hmm.
offspring,
Hmm.
not in the
Hmm.
mother,
Yep.
right? Because if you're being honest, it could have easily been a phenotype that's still occurring in you know not in those microglia. It could have been a
Right.
small
Right.
intestine issue. It could have been a liver issue and the periphery, et cetera.
Hmm.
And it turned out that those experiments where it's only deleted from the microglia completely reverse that eating
Hmm.
phenotype.
Hmm.
So those cells were completely able to eat.
I see. So all of the fructose induced problems that you saw in the developing brain, the failure to clear out all the gunk, required intact fructose transporters in the microglia of the little baby mice.
Yep, that's correct. I want to be clear that, there probably were also peripheral effects still
Yeah.
happen on the neonates, on those small mice. We just didn't look at those, but at least when you only hone in on the microglia phagocytosis defects, those were specific to the fructose. The effect of fructose was specific to them.
Mhm, mhm. And what's sort of like implied in a way by some of these experiments is that, you said you could get this phenotype even if there was no fructose exposure in the womb, but you just gave them, one tenth the dose shortly after birth. That starts to imply things like, oh, a small amount of fructose in, say, the breast milk of a human could potentially go in this direction.
Exactly. It implies that, I mean, if you don't want to like hope for a result, but I was kind of hoping that it was reversible because that would have
Right,
important public
right.
health implications. And it was not reversible. And so that was just pretty striking to me was, that those early hits make you susceptible. And we'll talk about what the ultimate behavioral phenotype was, but those early hits, they have their damage and it doesn't seem to be reversible just by stopping.
Right, right, right. And so, we see what happened at the cellular level? There's a phagocytosis deficit. Stuff is not being cleared out in the developing brain, in the frontal cortex in particular. This comes with just a fairly modest amount of fructose, either in the womb or even just after birth. What happens to these mice when they grow up? How do they look or behave differently than normal mice?
Yeah, so I think this is a really important question because, just because you have this cellular and physiological phenotype, you want to know if it's affecting behavior, cognition, et cetera. And it's hard to do in mice.